Hey there, you were hearing Franklin speaking with your host Shamanti, where I'll be in conversation with women in science, sharing their experience, contribution, and prospective planning in different scientific fields in an extremely entertaining and informative way. So stay tuned to find out some awesome woman centric science stories. I'll start this episode with one of Maya Angelou's saying, I would like to be known as an intelligent woman, a courageous woman, a loving woman, a woman who teaches by being. With all those traits and much more, our next guest, Miss Palina, is truly a phenomenal woman. Being through adverse circumstances, she chose her passion for science, embarked on research, and now influencing the world by her scientific contribution and artistic persona. So keep listening to Franklin speaking. I want to correct your pronunciation of your name. So is it Palina or Palina? I want to correct it.
Maybe the second. Both of it is okay. So you can call just Palina.
Palina. Okay. So you are a master's student at Nagasaki University, and
Yes.
you have done your bachelor's from Belarus, and you have also volunteered at UNICEF at Belarus, and you have also been assistant professor there at Belarus University. And alongside, you are also an illustrator and joining Nagazasshi magazine as illustrator. Yes. So you're thriving. You have different talents.
Thank you.
Yeah. So tell me about your study or what you are working on right now.
So right now I have moved to the new lab because I will graduate soon from my master course in September, so just next month. And I have finished all my work according to the master course, so I have defended my thesis and finished all the experiments and published the article. So just not to lose the time, I have already started the experiments in my new lab, which I have chosen for the PhD.
PhD. Yeah. And congratulations on
Yes,
acceptance to PhD also.
thank you very much.
And I've seen that you have also a paper published recently as a first author.
Yes, but anyway, all the courses significantly helped me in all this research. So I just listened to their recommendations and provided these experiments. But anyway, each of them made a huge influence on these articles.
So where are you originally from? Like I have seen a couple of places you have been through your academia.
Places of
Like you are from Belarus or you are actually from Japan?
I'm from Belarus.
Okay. So you are studying at Japan.
Yeah, so I'm Belarusian and I received my bachelor at my home country and moved to Japan just for this master course. So I'm living here for 2 years already.
Okay. So can you talk more about your research? Like what you were working on your masters and what you will be working on your PhD?
So as for the master course, the specialization was disaster and radiation medicine. We looked at the influence of ionizing radiation to human health or cells. So my department was the Department of Radiation Biology and Protection. So I was mainly concentrated on radiobiology. And as for the methods which were used in our lab, they were mainly cell biology methods. So I made conducted experiment and then looked into the microscope on cells to see how ionizing radiation, or in my case, in my research, I use just oxidative stress H2O2, and not ionizing radiation because it's similar to small doses of radiation. But anyway, I tried to irradiate cells just to see how is it going. So I treated cells with H2O2 or with some control chemical, and then looked at DNA damage, tried to find single strand breaks and double strand breaks, and then I looked at connection between these two types of damage, and suggested that because of the replication, single strand breaks are converted into double strand breaks. Yes, with different methods. There are specific methods to detect these SSBs and specific methods to detect PSBs, but all of it is through the microscope, and to have statistically proved data, I have to find at least 100 cells in a specific cell cycle phase, which I'm interested in. I was concentrated on S phase cells. And then to take photos or just to look at these 100 cells per one experiment and count either markers of DNA damage or calculate the intensity through the software, fluorescence intensity. So we mostly used fluorescent staining, and that's why I had to use fluorescent microscope, which is in the dark room, and you have to sit for several hours in the dark room and find cells. So it was rather difficult.
Yeah, I can understand. So it is a very interesting study. Like I have never heard of that SSB is converting into DSB in order to repair. I think it's the first kind of study you have done, yeah?
There are several articles suggesting this conversion, but these experiments were conducted, for example, on bacterial cells.
Okay, so you are doing human cells.
Yes, on human cells. So I think there is nothing about
like that. Yeah. So next, I wanted to ask you was what made you interested in working on this DNA repair or biomedical sciences as a whole?
So as for my bachelor degree, I studied at Medical University. So whole my education is on the medical field. And my specialization was preventive medicine. I tried to prevent diseases caused from different environmental factors, and one of the environmental factors was ionizing radiation. Because in Belarus we pay attention to it because of the Chernobyl disaster. And so it was one of the branches of my major. And so after that, so I have chosen Nagasaki University because there was a specific program. This is a joint program for radiation, joint between Nagasaki and Fukushima. It was created several years just after the Fukushima disaster. So it was very similar to the interest which I had in Belarus.
So is it true that there are lots of like people in Japan who had been infected, affected by the radiation and still have those kind of mutations in them? Like those nuclear bombs have affected that?
So we know that Japanese people were exposed to two atomic bomb explosures and to the Fukushima disaster on the nuclear power plant. But as for the health effects on them because of this radiation, it was proven that there is no effect after the Fukushima disaster because doses were very low. in comparison with Chernobyl disaster, where many people received very high doses and then severe effects were observed. In Fukushima, it's okay. Sure, as for the atomic bombing, there were effects on people, both deterministic and stochastic, proven. But now the dose is
Decline.
so everything is normal. Yes.
Okay, it is decline. Okay, so and
It's
then
normal, not normal city here.
okay, so
Because sometimes I tell somebody that I study in Nagasaki, and everybody knows Nagasaki because of the atomic bombing, and they are afraid that maybe there is radiation here. Now there is no radiation.
so
More than 70 years.
it has decreased. It is like normal now.
Yes, and everything was clean, sure.
Okay. So what would you say is the difficult aspect of the research you are doing in? Like the difficult things you are facing on a day-to-day basis?
For
Or
you mean
interesting, you can talk.
personally for me?
Okay, yeah, personally for you.
For me, it was difficult to work with a microscope.
Okay.
Yeah, it was very difficult twice. So I have to use eye drops to continue working because I have never done such work before. But anyway, it was interesting just because of this darkness, and you should concentrate on the moving fields. And I directly looked at the oculus of the microscope because because of the fluorescent staining, it was the most it was the best way to catch even the silent changes in cells.
Okay, yeah.
Then for me it was difficult to receive failed results, to have failed experiments. But I think you have heard from other researchers about this problem, but it was my first research, and I didn't know about the real situation, that in my master's thesis I will write just 10% of the results or 10% of the experiments which I conducted, and in the article maybe just 5% were written. Very small part. I have done so many experiments. Some of them were failed because I was on the process of training of the technique. Some of them were not used because then we changed the concept of the research a bit, the direction, so it was not necessary. And the most difficult for me was chromosome experiment, because here I had to receive well observed chromosomes. So the cells should be large and with well spread chromosomes, with appropriate distance between cells. In other way, I cannot take photos and then observe each separate chromosome in detail. So I even cannot obtain the result, and I have to retake the experiment more and more times, just yes, and slightly change the technique to find the way how to receive these chromosomes. I was thinking that I work with experienced scientists, they should know how to do it, but nobody knows what will work with my style.
Yes. So, these are the things, it happens, as we go on, we will understand that many more failed experiments are yet to come, I think.
I can say that failed experiment is normal. And if it is successful, it's something unexpected. So it should be such a statement in the scientist's mind.
Yes, that's great.
You should be ready to
yeah. So now I want to talk about what you are going to do next. You are going to PhD at the same university, like in Nagasaki?
Yes, at the same university.
Okay, so you are continuing with similar kind of research?
So the university is the same, graduate school is the same, but the department is different.
Okay.
So I was in the Department of Radiation Biology and Protection, and now I have changed the department to Neurobiology and
Okay.
Behavior.
Okay. So, from all the
So
best
as I
for your future research. All the best for your future research.
as I said, in Belarus, I had major in radiation sciences, and my master was in radiation sciences. But for PhD, I didn't I didn't choose the radiation sciences and switched to totally another field, because yes, before the masters, I was concentrated on the environmental factor which influences
Okay.
the human health. But as for the human health, I learned about it on different levels: on molecular level, cellular level, organs, in tissue organs level, organismal human level, and population level. So it is too unfocused study.
Yeah.
And when I tried laboratory work here, it was as I said, my first research work, just using hands, using these pipettes, tube cells. I really liked it, and I was thinking I would like to do exactly laboratory work. So I'm no more interested in the population level, in epidemiological studies, in statistical analysis of cohorts and something like that. So I'm interested just in cells and effect of it. But as I said, when you asked about the challenges, and I first said about the microscope.
Microscope. Yeah.
So I have chosen the lab where I will not use a microscope in such amount. And so it was a microscope is a method of cell biology.
Yes.
And now I will use molecular biology and biochemical methods. So no microscope, and moreover, I will enlarge my ability for laboratory work, and then after the PhD course, I have a wider range of skills which I have obtained. So I can do anything. As for the microscope, so I know how to conduct research using it, and if so I can design the research or even do it by myself or with assistant who will look for 8 hours per day to the microscope. But anyway, just knowing cell biology methods is not enough, I think, nowadays, because when you look at the requirements of different laboratory positions in various companies, you need to know everything, not just chromosome experiment, but also
Cell
PCR,
culture.
SDS page, yes, electrophoresis, Western blot,
All sorts
etc.
of things. Yeah.
Yes, all everything.
Yeah.
Uh,
Yeah.
because, for example, during master course, so I made I did a chromosome experiment, micronucleus assay, immunofluorescence. So as for the
All sorts
micronucleus
of
assay,
microscope.
many people even don't know what is it, and
Yes.
it's almost not used. It was just to support the results of immunofluorescence experiment. It's not so specific. So it's just roughly suggests the results.
Yeah, I just. So you are like broadening your horizon and learning at so much new things.
Yes, yes. But anyway, so I will work with cells, treat it with different substances
Different.
as I did
Yes.
during my master course, but just to use different other methods to
Yes.
detect change.
Yeah. So now I want to talk about like I have read that in Japan there is one in every 7 scientists are female. So you are probably going to be one of them, and you are inspiring so many people, so many newcomers. So uh, who would you say that your inspiration was growing up? Any professor, your friend, or anything? It will probably be a better if it's a woman, and who was your inspiration?
Inspiration to become a scientist.
Yes, yes, yes. Like,
Uh,
or in life, you can say both.
okay. So as for this fact about the women in Japan, so I would like to support it because in my new lab there are 6 researchers. Uh, 1, 2, 3, 4, 5. Yeah, 6 researchers, and only me is a woman. So 5 are
So yeah,
men, and only me is the
one.
woman.
So it's less than like
Uh,
20%. Like the percentage is mad.
yes, so only men before me were researchers in this lab. So maybe for them it's a bit strange, like for me too. But in Belarus there is just opposite situation because maybe it's not so well paid, and men prefer to choose some other career, and mainly women go to research positions at universities. Mm, so when I was thinking what to choose, so it went some some like smoothly. So I have chosen medical university because I was interested in chemistry at school.
Mm.
Then after studying at this medical university, I've chosen radiation. And then I studied, maybe just because I like to study. But everything was theoretical, and both in Belarus, as I said, I didn't conduct such real research. It was everything was theoretical. But here there was another type of research. It was real research in a wet lab by hand. Um, I cannot say that there is some exact person that inspired me to become a scientist. So, as I said, it went smoothly through my life.
Yes.
And I didn't know whom I will become, but now I'm here, and for that moment of my life, it's okay.
Yeah. So, like, you are probably an inspiration for many students back in Belarus and also in Japan. So, you are really doing great. So, I wanted to ask that there are, like, in India also, I see that the percentage of scientists, women scientists versus the male scientists are very like low. So what do you think is the reason, not in like as a whole in whole world, what do you think we can do, or the authority do to encourage more women or girls to be in science?
Mm, I think that this is because women have a dilemma in their life. Every woman has this dilemma to choose a family or to choose a science.
Career, yes.
Unfortunately, yes, it's a real situation. And if a woman decides to choose a family, it may become difficult to conduct research. Or as for my situation, my husband didn't support me in my research in Belarus because it was very bad paid, and so it wasn't enough. What was not enough for us, and he didn't support my inspiration, my passion for this work. So, but for me, the most important part is that I should love what I do.
Yes.
And not to work somewhere that in some place that I hate.
Yes.
So we had some misunderstandings about my work, about the place of research in my life. So, and finally we divorced because I have chosen science. So now I don't have this dilemma what to choose, family or science, because I have already done my choice. But as for other women, it may be so difficult to do it
Yeah. It's a very tough choice to
maybe
do.
they.
Yeah. It's a very tough thing to do. Like, kudos to you, but there are women who cannot really do that, or had doesn't have that much of capability or the mindset in them that they can, you know, uh, go out of a situation where they are being jeopardized their career and choose what they're most passionate about. So it's really very inspiring.
Sure, there are women that can do both
It's not
family
like that.
matters.
Yeah, they can if the family. Uh,
I think it's very strong women.
yes.
For example, in our university, I know just several women who are sensei, and yes, it's very inspirational that they can do both,
Oh,
to cope with their family and cope with their research and even medical practice as a postdoc.
yes.
It's very difficult for me. Yes, sure, it's very inspirational.
Yeah. Hats off to you. Yeah. So now I wanted to talk about your arts. You're really great at painting. I have seen many of your artworks, and I will put the pictures on the video. So the first thing I seen that when you first entered your lab, the reflection in your eyes, the conical flash, and that was like amazing. And the other one was like a DNA repair where a man was repairing the strands of DNA. So how did you get all those ideas? Do you like get those while working, or do you have to think about it?
Um, so I can say that I didn't paint too much before Japan. Just after arriving here, I started to do something like that. But I painted on general topics, and after starting the experiment, it really inspired me. And I really like these experiments. I spent a lot of time in the lab from the early morning till the late night doing this experiment. And then it was at home, and we had some New Year holidays, maybe I think something like that, around the end of December. And I was thinking, so maybe I would like to document some moments from my lab, because sometimes I'm thinking about the aim of painting is just to throw away the paper, and I think maybe it was paint something that I cannot take a photo.
Yeah.
So I can only paint it. And as for this first picture reflection from the lab, I have remembered the moment when I first came to the lab and I have seen all these micropipettes and the tubes and all the lab equipment, all the dishes. I was so impressed because, for example, in Belarus, I have seen the micropipette only once in my life, and just to look at it, not to touch, and so surely not to use. And here I was allowed to use everything, and I was taught how to do it, and I could do experiments using all of this equipment. And for example, as for the dishes for cell culture, they were plastic, and I could throw it away just after one use, and not to autoclave and reuse it again and again. So it was really impressionable for me, and I really looked with such eyes, with large eyes, and trying to catch all the details here. And it was my first lab, and I looked the first days I looked at everything, and then asked and say, what is it? What is it? It was not connected to my research. It was just something that played on the bench, and I was coming to him with a list of questions, and he thoroughly answered all of it. Now in the new lab, I know most of the things, and I'm not so excited as in my first lab. Yes, but it was funny, and I made a series of these pictures about my laboratory, and then I came to my professor of the lab and showed it.
Oh,
So he was really laughing. He didn't expect it, first of all, and it was showing all my emotions from this
yes, yes.
laboratory. I was like a child here.
Yeah, you were
Yes,
amazed. Yeah, I can understand because when in bachelors also, here also we are not allowed to do much. So when I entered the masters and entered my first laboratory, I was also okay. There are so much things to do, and like I haven't read enough of this, don't knew enough of this. So yeah, I can see it's a great
yeah.
experience. So another thing I wanted to ask that arts and science doesn't really, stereotypically doesn't go hand in hand. There are so many stereotypes that when you are doing scientific research, you have to, sometimes compromise on your artistic side. But you are doing so great doing both simultaneously. And I also think that it's very important to keep your artistic creative side on mind because sometimes it, you know, creates revelation in your research also. So what is your plan regarding your arts? Like, do you want to sell it or exhibit it somewhere or anything else? Will you be continuing it while doing PhD?
I would like to continue doing art because I really enjoy this process. But I don't know if I will receive some support in my new laboratory because I think that here the lab is more serious, and I think if I will talk about my art, they will just say that I'm wasting my time. I should do more experiments if I have time.
Okay.
Um, so in my previous lab, it was just a master course. It was maybe not so serious in comparison with a PhD, and yeah, they supported me a lot with these paintings. And as I said, I showed it first to my professor and supervisor. Um, this lab, maybe I can feel even by myself that I wasted my time and I should work more on the experiments. In this case, I plan to think more about the more serious scientific illustrations, for example, to create artistical chemical pathways, for example, for my hypothesis and for the results. Anyway, I will do it because as for the master course, I included such illustrations for the presentation for thesis defense. So anyway, it is necessary so I can put my inspiration at least at these illustrations, and nobody can tell me that I was wasting my time
Ye-
because
Yes,
it
you are doing your work.
contains. Yes, I'm doing my work and it contains information that was necessary to include to some slides. And as for the exhibitions, I don't know. So I tried to organize an exhibition in my university, but because of the coronavirus, it was prohibited, and I think it will be prohibited for a lot of time even after that because of the pandemic. So my professor just put my artworks on the website of the laboratory. So it was
Yeah,
like
I saw that.
something online exhibition. Yeah, so I think it's enough for me. And then this Arts and Science series was published in Nagasashi
Yes.
on the summer issue. So I think it's enough for at least for this series. I have started the series of this new lab, so I will know more paint about the master's lab. But as for the PhD, I have just three, yes, so three paintings. But maybe it's just for me. When I look at it, I just remember these moments because I don't paint just something to paint. I have some situation, emotional situation, which I just put on the
On the
paper.
painting,
Uh,
yeah. You're really living
yes.
through all those paintings.
Uh, Yes, if I had some stress, it's not just about happiness. It's about
Mixed in it.
stress, fear, danger, or something like that. Anything.
It's great. I can see the paintings behind you also. Those are really great. Like I haven't seen something like that before.
This is from my first lab, like
It is
washing
at
dishes.
washing, yeah. I saw it on your Instagram.
Yes, for me it was unexpected. In Belarus, we scientists don't wash anything. There are some laboratory assistants who do it, but here in Japan, I think it's because of the culture, because everybody clean his own workspace, and it doesn't matter if you are professor, associate professor, or just student. Everybody clean.
That's very important.
Yes, and after experiments, everybody washes everything after usage.
Clean up after yourself. Okay, so I guess it will be the last question of this podcast. I wanted to know that what would you say to the upcoming, women or girls who are coming into science, either in like Belarus or Japan or in a world, that what do you want to say? You can say that in your own native language also. I will appreciate if you say that.
Um, so I would like to say, because I was faced with such situation, I was thinking that my research is not important. And I was talking a lot with my sensei, and he said even a small part of it will be published, and other researchers can look at it, and maybe they will decide not to repeat it because it is already done. So it will save the time of other scientists, or maybe it will be used in other research to support some idea. It's always necessary.
Unless friends,
So
yeah.
don't think that if you didn't develop some medicine for cancer treatment, it doesn't worth to be done. Sure not. So if you want to do something new, if you are curious about new knowledge, so you have this scientific curiosity, please do at least something. It will will be valuable.
Yeah, that's really great. And anything you want to say in your own native language, because I want to like incorporate diversity, because so many people from so many different countries are going. So can you say something?
In my native language, so in Belarusian,
Yes.
I would say Белорусы, тримайтесь, и все будет добро.
What does it mean?
So I just said that Belarusians should keep the spirit, and everything will be okay. And so, yes, because of the situation in our country nowadays.
Yeah, I understand. Yeah. So thank you very much for joining this podcast. You've been a great for this podcast, because you are a very experienced and inspiring person, and I hope all the best wishes for your upcoming endeavors. Thank you so much.
Oh, thank you very much. You asked me so many interesting questions, and when you asked again and again, I came up with some ideas which I didn't think before. So it's really a talent to ask questions. I think you are doing really well. Thank you very much for this interview.
Thank you. So I will just thank you so much for tuning in. Palina's artwork and her paper will be linked in the show note and description below. Do check those out and keep listening to Franklin Speaking.